The bells are ringing again, and this time with Professor Yonath, the 2009 Chemistry Nobel laureate with us. Bells, Berchmans Erudite Lecture Series, is SB's signature program developed by the visionary teachers of the college to commemorate the 90th birthday of the college. It was inaugurated by Professor Ishi Negishi, the 2010 Chemistry Nobel laureate. Bells aims to invite scholars of excellence of various disciplines, especially Nobel laureates, to the campus for lectures and interactions with the students and faculty. This time, Bells brings Professor Yonath to the campus.
Professor Yonath, lovingly nicknamed as a lady with head full of ribosomes, was awarded this Nobel Prize for the crystallization of structural ribosomes and their structure elucidation. Dr. Ada is a professor at the Weizmann Institute of Sciences in Israel, and she is the first one to establish a biological crystallography lab there. Professor Ada was born in Jerusalem in 1939, and she was born to a poor family, and her firm determination and perseverance made her achieve this Nobel glory.
Professor Ada delivered the Bells lecture on 5th February 2019 at Kavakot Hall. The topic of her talk was From Basic Science to Next Generation Antibiotics.
Proteins exist in every living cell, in bacteria, in chicken, in cockroaches, in fish, in flowers, and in people. And they perform almost all the tasks that are needed for life. You have here an example, or several examples, of proteins, human proteins, like the proteins that are structural in the hair or in the skin, proteins that are regulating, like insulin, that regulates the sugar level in the blood, transport proteins, those that transport the oxygen from the lungs to wherever they're needed. This is a protein, a red protein called hemoglobin, sensory proteins, and enzymes. Enzymes are the proteins that are chopping down the proteins that we eat in order to make the proteins that we need. So if I can repeat, proteins perform almost all cellular functions. Those that are needed for smooth life, like breathing, digestion, and those that are needed for pressure or problems, like heat shock or cold shock, or immune response, organ failure like heart and so on. In nature, there is a law. In nature, structure means function. You can see here the two examples of a very, very complicated machine, paper clips. Then they can do their job only if they have the right structure. If you change the structure even a little bit, not cooking, not boiling, not cutting, not freezing, just change the structure, they cannot do the job. And that's correct for every molecule in life, in nature, not only in life. So proteins are long chains that are made of amino acids. There are 20 types of amino acids, and the fold of each protein, which is carefully designed for fulfilling its function, is determined by the sequence of the amino acids, and this is determined by the gene. Each protein has a gene. Each gene is a coding for a protein, and the sequence of the code in the gene determines the sequence of the amino acids. It means the structure. All the genes are in DNA. In order to make them useful, the DNA has to change itself, to open itself. It cannot. It's a double-stranded double helix. It means here the instructions are covered. So first of all, it's being transcribed to a similar molecule, and both of them have the whole language, the whole biological language is 4 letters. Both of them have 3 out of the 4 exactly, and one a little bit different. So here is the information that can be translated by ribosomes to proteins. That's the story. There is DNA, there is ribosome, and ribosome produces the newly born proteins according to the DNA. So ribosomes are universal. They function in almost the same way in all living cells. Doesn't matter if the cells are small and ancient or new. And there is a huge number of ribosomes that function in each living cell. In mammalian, in us, like in our liver, can be 4 to 5 million ribosomes in one cell. Even in bacteria, about 100,000 when bacteria grow. They work very fast in vivo in the cell. They can make up to 40 bonds between the amino acids that make the protein. They are called peptide bonds in 1 second. And they hardly make mistakes. Mistake rate is about 1 to a million. So I was a good student and a very fast student when I studied chemistry. You heard already that I came from a poor family, so I had many jobs. This is why I was fast, not because I was very good or so, but because I had many jobs and I had to come to the jobs on time, so I finished the lab as fast as possible. Our exercise was to make 1 bond, and I was the first. I needed for 1 bond 6 hours, and they can make 40 in 1 second. Very talented cellular components. Now that we know how they work, we like to describe them as a factory that has 2 floors. In the top floor, the instructions are being read, and in the lower floor, the bonds are being made. So when here, a triplet, each amino acid has three letters that code for it, as I said earlier. When triplet of letters come here that are coding for that amino acid, it will get in and be connected to the newly grown protein inside. This arrow has to be inside. Once this happens, the protein is a little bit longer, one amino acid longer. The message progresses by 3 nucleotides, by 1 code, and the track, together with the factory, are ready for next amino acids. The track is also a molecule that has 2 sides. One side can read the codes. It's called anticodon because it makes pairs, like in DNA with the code, and the other is carrying the amino acid. So this is the tracks, it means tRNA molecules. Once we got the structure, we were so excited because we also understood the function. And we made little videos that can be found in, oh God, it doesn't work. Can be found in YouTube. Yeah, it works. So what you see here is the messenger reaches the small subunit, the top floor of the factory, finds its way into its path, and it's all controlled by factor 3, which is the blue thing. Once this is okay, the first track is coming, the first tRNA molecule, binds to its position, brought by another factor, and now everything is fine. The large subunit can associate with the small one, and the active ribosome has been made. So we look at it from the solvent side. Yeah, okay. Below is the small subunit, above is the large, and you can see how from the left side amino acids are being brought by elongation factors, these blue things. They are decoded and connected, the amino acids are connected to the newly born protein, as you can see here. We took away the large subunit so you can see the decoding. And the lower part, you can see that the newly born protein goes into the tunnel that protects it, and this tunnel is in the large subunit. Now you can see the whole ribosome, and proteins come out from the lower part. This protein is now coming from the lower part after the tunnel and can fold the way it's needed. The whole process continues until there is a stop codon, and then instead of tracks is a factor that is release factor or recycling factor. It comes in, it separates the two subunits, the protein goes out to where it's needed, and the rest of the particles, the rest of the players wait for the next job. So that's the whole story. And what actually you saw in the movie or or heard or read, two subunits, small and large, they get together through the tRNA that is doing decoding here, and peptide bond is being made here. And you can also see it if we look at it this way, small subunit, large subunit, decoding here, and building the protein here. And until now, I didn't show you very accurate structure, so I just want to show you that we do have, and you saw it also earlier in the introductory lecture, small subunit, large subunit, made mainly of RNA, again RNA, ribosomal RNA, and many proteins, each protein in another color, whereas the RNA is gray. So I hope that I convinced you that ribosomes are important, and because of the fundamental role played by the ribosomes, many antibiotics stop them. So if you want to stop the life, what is this? If you want to stop the life of a bacteria, of a bad bacteria, pathogenic bacteria, stopping protein biosynthesis is a good idea. It will not be able to exist. So almost half of the clinically useful antibiotics are targeting protein biosynthesis by paralyzing the ribosomes. Did you know that the natural antibiotics are the weapons of bacteria, one bacteria against another type? So if you didn't know, now you know. These are natural products. Small antibiotics, very cleverly designed. A chemist should appreciate it very much. And the first question we asked is, how do the tiny antibiotics paralyze the giant ribosome? So antibiotics molecular weight is less than 1,000, and ribosomes are 2.5 million. How do these little ones paralyze this large one? And the trick that they are playing, they are binding to active sites. So you can see here the small subunit, the skeleton, and the large, and each circle represents an antibiotic. And if you look where they bind either in the decoding or in the bonding place or in the tunnel or in the hinging, these motions that you saw in the movie. So we now know how the antibiotics work, much more than I expected. And when we got the structures, we immediately understood how this happens. And we made another video, which I want to show you. Both videos we made with two students of the Art Academy. So have a look. This is a very small antibiotic called edein, and very clever. Small but clever. Exactly in the path of the messenger RNA. You don't have to be big in order to disturb. Second one is also a small one, all of them are small, and it occupies the position of the second tRNA, the second track. Just occupies the position. No binding, no bonding, no protein. The third one that I want to show is blocking the tunnel in the large subunit. It's called erythromycin, the first one of the ribosomal antibiotics that went into use. See, just blocking the tunnel. Protein can be made but not go out, and only very little protein can be made, as you can see. And the last one that you will see, it comes in a minute, is the most clever chemically. Goes into the bone. Antibiotic goes into the bone. It's called clindamycin. You remember where the bones are being made? It just goes there, sits in the bone. So these are the natural antibiotics that are being used today. And what we learned about them, all ribosomal antibiotics bind to ribosomal functional sites. But I said earlier, all ribosomes work the same way. It means ribosomal functional sites are highly conserved. And it's very important, mandatory, that we don't kill the patients on the bacteria. So how does this happen if the sites are so similar? By small changes, which I will not show you now, but there are sometimes subtle changes between the active sites of the bacteria and of the patients. So this problem is more or less solved, but the next problem, resistance, is a real awful problem today. More and more resistance to antibiotic is being made, is acquired by the bacteria. And before I go on, I just want to tell you that even the resistance is a natural product of bacteria, the bacteria that is being hit by the bacteria that hits it, and the resistance, the finding that is natural product, and the resistance is really alarming. And I am afraid that we are reaching a period, as it was before antibiotics, that people died from simple diseases, even like wounds. This, together with the fact that big companies stopped making new antibiotics, stopped looking for new antibiotics, gives me the feeling that we are going back to pre-antibiotic period. As I said, people died even from wounds, or as the World Health Association said, post-antibiotic area. But anyway, antibiotics will not be useful if resistance goes on. Even the World Bank estimated that almost 4% of the global economy will be lost by resistance. And as I said, yet companies don't make it. So I became very worried about it. And first of all, I thought, is it possible to fully combat resistance? Can we do it? I think it's unlikely because bacteria want to live and because they are cleverer than us in their wish to exist, in terms of survival. Nevertheless, as I said, most pharma do not do it, and soon people will die like these giants. All died before the age of 50. Some of them even before the age of 40, like Mozart and Schubert. All were giants. All died from diseases like infectious diseases, and the companies do hardly anything. So this is the way the growth of resistance of 3 different antibiotics, and this is the amount of new compounds, new antibiotics. Now, hardly anything. In the last 20 years, only 3. So we decided to do something about it. We thought that if we understand the structure of the ribosomes, of the pathogenic bacteria, it means the bad bacteria, maybe we can find regions that are different from the good bacteria, and we determined the structure of several pathogenic ribosomes and compared. And we learned that all ribosomes are similar. We also collaborated with a company that could make antibiotics better, like Nabriva in Vienna. We also found regions that may be used for making the normal antibiotics more selective. But the real results that excites me is that beyond my expectation, we discovered novel binding sites, new binding sites, and I want to show it to you. So what you see here in gray is a normal ribosome, ribosome from a normal bacteria, and the cyan pieces are those that are found only in the pathogen. Not many of them, less than 10%, but all of them that I show here have structure, unique structural motif. Have a look here, for instance. Let's focus on one. See here, they are exactly the same, the pathogen and the non-pathogens, also here. But here there is a little extension of the pathogen, and this has a structure. So maybe it can be a potential binding site. So altogether, in one bacteria-specific Staph aureus, we identified 25 positions. We blocked each of them alone by chemistry, and we found that 16 of them, when they are blocked, they inhibit protein biosynthesis. So this is a great finding, because these sites are not involved in the primary ribosomal activity, and therefore there are no genes for their modification. This means that for now, we can think about exploiting them for new antibiotics. So before I go on, I just want to tell you that our new antibiotics, except for being new, so less or no resistance so far, we want to make degradable and environmental friendly. So about degradable and selectivity, let me just talk about a minute about the microbiome. I hope that you all know about the microbiome. These are the millions of good bacteria that grow in our body, and they all can be destroyed by antibiotics, the normal antibiotics, if the antibiotics reach them. So a person can get antibiotics against cold and come out with a real bad immune disease. So we want in our new antibiotics to preserve the microbiome, not to damage it. Also, we want to be environmental friendly. So most of the ribosomal antibiotics known today have it between them, inside them, pieces that are not degradable, not digestible and not degradable. So they go to the soil with all the other things we don't eat, we don't digest. And because they are so small, most of the purification systems don't catch them, and therefore they can penetrate into the irrigation systems. And therefore they can be used for growing grass or fruit or vegetables, and we get them back. And this even increases the resistance. So in our new positions here, that I showed you one of them, we can design the antibiotics so they are, from the chemical point of view, they can be optimized. They can be optimized in terms of properties. They can be optimized in terms of antibiotic action and degradability. So in summary, the new insights obtained from these high resolution structures of the ribosomes from genuine pathogens provide unique chemical tools for suggesting novel sites for antibiotics for suggesting how to improve. I didn't show, but I told you, or how to distinguish. And this is what we want to do. We want to make pathogen-specific antibiotics. For each pathogen, its own antibiotics. This will be also leading for less usage. So this is what we want to do, what we're doing now, and we want to do in the future. I also want to tell you some words about how we started, because we heard most of it earlier. So we started with crystallography, because only when one knows structure, one can understand function. And at that time, in order to see very fine structure, very accurate structure, one needed crystals. So I will not explain crystallography now, but it is based on diffraction, on X-rays, Röntgen rays that diffract from a periodically packed object, like here. X-rays come in, there is here a crystal, and the X-rays are diffracted, and we can see the diffraction. The further there is diffraction from the center, the higher the resolution. Or if we want to describe the whole system, crystals are being hit by X-rays. They produce diffraction. We can collect it on film or on detector. We have to collect and combine all the diffraction, millions of diffraction spots. There are some ways to do it, and the crystallographers, like this little man, should know it. And from this, one can suggest the structure. But crystals, here is a crystals of sodium chloride. It means salt, the table salt. It's made of 2 atoms, and they really like to make crystals, even spontaneously, fully periodic from 3 sides, height and the 2 x and y axes. This is possible. It's also possible to crystallize proteins. Not so simple, but it's possible. But these guys, the ribosome, when I started this work, it had a big question mark, which actually said no. And as you heard earlier, many people doubted it, but they doubted it because they tried and they could not. You know, I was not the only one that wanted to understand how ribosomes work. Many wanted, and they were more mature than me, more established, they had better labs. I was just a relatively fresh young scientist, and they failed. And they explained the failures so bad, the failures were that ribosomes were considered non-crystallizable. They explained it by high degree of internal mobility, flexibility, and so on. They were right, but they overlooked one of the reasons that I thought is most important, and this is the marked tendency to deteriorate. So when I prepared ribosomes, it doesn't matter which method I used, I got half of the activity that I expected, and I asked all the leading ribosomologists in the world, they also got even less than half of the expected activity, and they thought it's good. But I thought it's bad. I thought that if we get less activity, it means some of the ribosomes are deteriorated. And if in my container, glass or whatever it is, I have several types of ribosomes, fully degraded, half degraded, partially degraded, not degraded. How do I expect crystals? This is not homogeneous. So I thought that this is the most important reason. I didn't really know how to solve it, but I had luck. I had a bicycle accident. You could also see it earlier. And while recovering, we took 11 months, I read all types of things, not only science, and also about the bears that go for their winter sleep. I read about a delegation that went to see how they metabolize when they are sleeping. So they found lots of things. One thing that they thought is not so important is that the ribosomes are fully ordered and very well packed in the inner side of the membranes of their cells. All of them. So this, first of all, showed that it's possible to order ribosomal particles. People could not, but every bear, every winter, every cell, it's a mechanism that nature provides that makes it possible. And I thought, why does it happen? So in my opinion, it happened because when the bears get up at the end of the winter, they want to do all types of things. They need proteins. For this, they need ribosomes to make the protein, so they have to preserve it. It means stress induces periodic packing of ribosomal particles. Stress doesn't have to be cold. It can be also heat. So I wanted to use ribosomes from bacteria. I didn't want to go to the North Pole and kill bears in order to understand ribosomes, kill bear after bear. I wanted to use bacteria also because they are much more important medically. And I used ribosomes from bacteria that live under stressful conditions like the Dead Sea or hot springs or waste of nuclear facilities. So the Dead Sea is the lowest part in the world. It's in Israel. You can see it here, a piece of it from top. No animal, no tree, no nothing. When there is a lot of evaporation, it gets these structures out, but these are all salt. Because the salt, it's not only the lowest part in the world, it's also very hot. In the summer, it can reach 60 degrees Celsius. It's even hot for India. And it's full of salt. So salt, when water evaporates, salt comes out to the surface. So for years and years, it was considered being Dead Sea. But two professors of the Hebrew University in Jerusalem found colonies of bacteria. You see this pink line here or here? This bacteria is called Heloarcula marismorti. Helo because it's full of halogen. Arcula is the type of the bacteria. Marismorti is Dead Sea in Latin. So I thought about using the ribosomes from here or from here, which is a bacteria that lives under every condition, very hot, very cold, no food, hunger, and even irradiation. It's called Deinococcus radiodurans. So we started crystallizing, and within half a year we got very small crystals that were made of ribosomes but were still not ready. But in 4 years, they grew beautifully, and we had several types. We even improved those, and we went to measure them. And measuring crystallography result is by using very intense, very intense facilities like this accelerator called synchrotron for X-rays. This one is in France. At that time, there were only 5 in the whole world. In Hamburg, in Cornell, in Stanford, and in Japan. So we went there, and let me just show you how it works. So particles are being accelerated here, and then run around in this tunnel, the big one, and in the tangentials there are stations that we can measure. Here, Frank from my group is measuring. So 6 years after we started in 1986, we got the first picture. Beautiful, look at it, with spots to the edge, with highest accuracy. You can imagine the students, wow, we have, yes. Not for long. Second picture was already gone. So we repeated this experiment 100 times in all these 5 accelerators, 5 synchrotrons, and we got the same. Sometimes we got 2 pictures, but not more. And this is less than 1 promille of the data we needed, and collecting it from 10,000 crystals looked completely impossible. So how did we feel? You heard already. We stood here in the beginning. We stood here and we saw only this mountain. We called it the Everest. So we started climbing it, climbing and climbing and climbing for 6 years until we got to the summit. And what did we see? That there is a larger, more difficult Everest in front of us, the Everest beyond the Everest. So this was really time that I thought stopping. But before stopping this project, I thought, why does it happen? And in my opinion, I thought that all the crystals, all the tissues, all the biological tissues are damaged by X-rays, but ribosome crystals most, because the damage happens in two steps. Primary, that the X-ray beam breaks the bonds. This we cannot stop. But then the bonds, when they are broken, the two atoms of the bonds are unhappy. So they run around to make themselves happy and break more bonds because they want the electrons out or in. And this running around is easier in ribosomal crystals because they are so soft, and this can be stopped just by taking away the energy for running around. It means by working at almost 200 degrees, minus 200 degrees Celsius. So here too, everybody told me it won't work. We tried and it didn't work. They didn't try. All those tried before us tried minus 50, minus 80, but not minus 200 or around it. Yet, we decided to it took us a year to make sure that we know how to flash freeze the crystals, and the experiment was done in Stanford together with Hakon Hope. Look at my face. I don't look here that I'm really expecting, but it worked. And this was in November 1986. By March 1987, it became routine in all the labs, first of all in the synchrotrons. First of all in synchrotrons, but then in labs. But before I go on, I want to show you how beautifully the pictures look, the diffraction to the edge, and we can collect almost full data set from a single or two crystals. You can see that companies started to make the machinery for flash freezing. Here there is one installed in Grenoble, and look at my face 20 years later. I'm much happier than in the day that I suspected. And look what happens in the world. So if you look at the number of structures, which is shown here, and the year, when we started, there were less than 100 structures of biological compounds. And then, look at it, within a few months. Now, there are, in 2011, there were about 47,000 new structures. Now there are 89 new structures. And all of them in our system, students don't even know that there is other systems now. And all of them of materials that are either important medically or for new materials or for understanding new processes. So this is the story that you heard earlier, and I want to finish by thanking the Weizmann Institute that let me work, although I was called the dreamer, to Professor Wittmann from the Max Planck in Berlin, that all what he wanted is to see whatever possible with ribosomes, so the collaboration was really fantastic with him and his group, and he was very important in starting a research unit for us in the synchrotron in Hamburg. So essentially I had two groups working in parallel, one in Hamburg, one in Israel. He died 10 years before the structures came and was replaced by Franceschi and then by Fucini. So I also want to thank the members of my two groups, the one in Hamburg that was closed 15 years ago, and the one in Israel, for their devotion and determination. And I want to show you the one that is still functioning in Israel. So you can see my face here, but actually the senior scientist is Dr. Anat Bashan. And for the young female here, it's possible to be an excellent scientist and fantastic mother. She has 3 children, they're all wonderful. Ella is also an excellent scientist. She has only 2 children, they're wonderful. Raz is excellent, he's now a professor. He has 3 children, but he doesn't belong. We talk about female now. So let's talk about Tamar. She came 16 years ago for 10 weeks and stays until now. Now herself has 3 children. But the day that I took the picture, she baked a cake. It was her birthday. I want to show you, female can even bake cakes, good scientists. You see the cake here? Maybe not clear enough, so I enlarged it for you. Here it is. And this shows that in my group, ribosomes are considered sweet. I also want to thank my family, my daughter who is an MD and granddaughter, and my sister and so on. I only showed the 2 of them. And I want to tell you that my granddaughter, 11 years ago, gave a speech about me when I got a prize in Paris. And she said, as you know, she, it means me, is a very busy scientist, but she always finds time for me. And she gave me a prize. That's not her prize. This is the Nobel medal. But for me, her prize is at least as important. The grandma of the year. The grandma of the year is Yonath. And when I asked her which year, every prize has a year, so she said, you have to reprove yourself every year. The minute you fail, I take it off the wall. I want to show you it's still on the wall. I also want to show you that ribosomes became popular in Israel. This is a carnival. He is me and his wife. Look, they are dancing. She is the ribosome. A protein comes out of her. Even young children became ribosomes. And this is the way that I was described by one of our best artists. You heard it earlier. Small subunit, large subunit, and he thinks new antibiotics should be here. His name is Michel Kishka. Thank you.